Hi and good day to my lecturer. My name is Dayanur Amira binti Abahalim Badasham and I'm going to present my poster of my mock research which is entitled Antimicrobial Activity of Gingiber Officinale Extracts Against Foodborne Pathogenic Bacteria. This is how my poster looks like. I use a picture of Gingiber officinale or ginger as the background for my poster to symbolize the title of my mock research proposal. Foodborne illnesses are usually caused by different species of bacteria and their toxins, viruses, chemicals, and other microorganisms. Foodborne illnesses are only transmitted to humans through the intake or eating contaminated food, such as raw meat products, raw milk, raw or uncooked vegetables, and contaminated water. Examples of foodborne pathogenic bacteria are Escherichia coli, Salmonella species, Staphylococcus aureus, Vibrio cholerae, and Klebsiella species. Antibiotics are one of the antimicrobial agents that used to kill and destroy the growth of bacteria. It is also used to treat many diseases, including foodborne illnesses. However, if antibiotics are highly consumed, it will cause many other side effects such as clinical bleeding, drug rash, acute pulmonary reaction, and vomit. Thus, an alternative medication which is easy to find and less side effects is needed nowadays. It is the problem statement for my mock research proposal. Gingiber officinale or ginger is used widely all over the world as medicines and antimicrobial agent. Ginger has long been used to relieve food poisoning, cramps, sore throats, vomiting, flu, and indigestion. As we can see from the picture shown, ginger has been used as ginger oil and ginger water. Hence, this study was conducted to determine and identify antimicrobial activity of ginger extraction against foodborne pathogenic bacteria, such as Escherichia coli, Staphylococcus aureus, Salmonella species, Vibrio cholerae, and Klebsiella species. Soybean oil method has been chosen over ethanol method in extracting ginger in order to evaluate the antioxidant activity. There are two hypotheses of my mock research. Firstly, ginger may be an effective antimicrobial agent in killing foodborne pathogenic bacteria and treating various foodborne illnesses. In addition, antimicrobial activity in boiled ginger extraction may give a higher and more effective than in soybean oil alone. Next, I will talk about the objective of my mock research. First and foremost, this study is studied mainly to identify the antimicrobial activity of ginger extraction against different foodborne pathogenic bacteria. Furthermore, extracting ginger by using soybean oil at the boiling temperature is one of the objectives of this study. Lastly, it is also aims to compare the antimicrobial activity between in-boiled ginger extraction and in-boiled soil alone against foodborne pathogenic bacteria. What I can expect from this study is antimicrobial activity of ginger extracts may vary based on the bacteria species, as shown in the figures. Apart from that, I also can expect that boiled ginger extract, which is soybean extract of ginger, may give a higher antimicrobial activity against foodborne pathogenic bacteria than in soybean oil alone. Last but not least, Salmonella species may show the highest antimicrobial activity of ginger extracts, while E. coli may show the lowest antimicrobial activity of ginger extract. Staphylococcus aureus, Vibrio cholerae, and Klebsiella species may not show the highest nor the lowest in antimicrobial activity. There are 6 main methods that we need to focus on in this study. Firstly, ginger and soybean oil, as shown in the picture, will be purchased from a local market. 5 bacteria species will be isolated from food sample and will be used in this research, which are Escherichia coli, Staphylococcus aureus, Salmonella species, Vibrio cholerae, and Klebsiella species. We must not forget to preserve these bacteria. In the second main method, which is inoculum preparation, first and foremost, subcultured bacteria will be inoculated into tryptic soya broth and shown in figure 4 and incubated at 37 degrees Celsius in order to measure the turbidity of 0.5 McFarlane standard. In the figure 5, we can see the example of inoculum of bacteria onto the Mueller-Hinton agar. For the third main method, which is media that will be used for this diffusion assay, is Mueller-Hinton agar. Twin 20 also will be added to the agar. The picture here illustrates before and after the diffusion assay. In preparation of ginger extract in soybean oil, 40 ml soybean oil will be mixed with 10 g ginger powder. Then it will be boiled for 30 minutes and after that it will be cooled to room temperature before being filtered through hot filter paper. The extracts will be stored at 4 degrees Celsius before being utilized in the diffusion assay. Next, I will proceed with the fifth method, which is antibacterial assay. Filter paper disc with 6 mm of diameter will be prepared and a lot of each bacterial isolate will be made on Mueller-Hinton agar plates. Filter paper discs will be stacked one on top of the other and ginger extracts will be added to each disc separately. The antibacterial activity of soybean oil alone will be compared to that of boiled extract. Gentamicin this will be utilized as a control. For the last method in this study, which is statistical analysis, we can determine the antimicrobial activity of all those 5 bacterial species by measuring the zone of inhibition diameter of the agar surface after 18 to 24 hours of incubation. To conclude, ginger plays an important role in treating diseases and destroying bacterial growth since it is one of the largest plant sources that can be used as antimicrobial because the presence of active compounds in ginger such as gingerol, and paradol. Apart of being antimicrobial agent, ginger also has been used as antioxidant, anti-inflammatory, anti-tumor, anti-diabetic, neuroprotective effect, and hepatoprotective effect. That's all for my presentation. Thank you.